Good afternoon, class 11th. Today we once again continue with the second chapter in biology of biological classification. And in this chapter, children, in video 17, which is the last video, children, we had studied about sporozoans as parasitic protozoans. We studied specifically about malarial parasite. We studied about the various cycles of malarial parasite Plasmodium in the primary host and the secondary host. We studied about ciliated protozoans and we saw their characteristics and also the specific example of paramecium, and we studied its characteristics. Today in video 18 we will do a run through or a quick review of fungi, although not in course but for competitive point of view we will go through this. These are heterotrophic organisms with over 1 lakh species, from unicellular yeast to the toadstools to puffballs, etc. They have habitats, both aquatic and terrestrial kind of habitats. And the fungi are eukaryotic, are heterotrophic. They obtain their food by absorption rather than by ingestion, and they give digestive enzymes outside their body and then absorb the products of digestion produced outside, as we can see here in this diagram that from outside they give in the mycelium or the hyphae inside, and these are special hyphae, and these then extracellularly they do the digestion inside the host. So we see that they give digestive enzymes outside their body and then absorb the products of digestion that are produced outside. They suck it back into their hyphae. And we see that the fungi are studied under the botany branch, which is known as mycology. Let us look at some of the fungi that are shown here, children. Let us look at some of the fungi, and we see that the fungi, they are studied under mycology, and we will look at the status of the fungi. They are eukaryotic, they are spore bearing, as we can see here in Rhizopus. We can see the spores. They are spore bearing and they reproduce both asexually and sexually. We see in penicillium the conidia that are given here, then in Saccharomyces, in Polyporus, in Peziza, and in Agricus. We see that there are different kind of fungus here that have been depicted throughout these various diagrams, and these are the various forms of fungi that we are looking at. And we see that Copeland in 1956 he placed the fungus with algal and protozoan groups in kingdom Protocyst, whereas Whittaker in 1969 he raised them to the status of an independent kingdom. And we see general characteristics of the fungus. Here the fungi are eukaryotes, they lack chlorophyll, and that's why they are heterotrophic in nutrition, because they lack chlorophyll, and they are unicellular yeast, and the other fungi are filamentous fungi. They have branched thread-like structures called the hyphae that we can see here, and these hyphae, they form a big net together which is known as the mycelium. The hyphae, they make a net together. And we see that the hyphaes are both aseptate, whereas the septa or the divisions, they are not dividing the hyphae in between, and they are septate hyphae also, which are divided by septa in between. And the aseptate hyphae are also known as choanocytic hyphae. And we see that the mycelium of the parasitic fungi have hyphae that are specialized, and these specialized hyphae are called haustoria. And we see that the hyphae wall is made up of chitin. Do you see this hyphae wall? This wall is made up of chitin, a nitrogen containing polysaccharide material. And we see that the cytoplasm has all organelles in it. The organelles in the cytoplasm, they are mitochondria, Golgi apparatus, endoplasmic reticulum, ribosomes, and vacuoles. And the fungi have an assimilative or vegetative and reproductive phase. So this is what we have seen in the general characteristics of fungi. Then we come to the nutrition in fungi. As I told you, they are heterotrophic in nutrition because they lack chlorophyll, and they require organic source of nutrition in the form of carbon, nitrogen, amino acids, and organic sources. They want inorganic sources of nutrition are required by them. Ions like potassium and magnesium ions are there. Traces of iron, zinc, and copper are required as nutrition by fungi. We see they are obligate parasites also. That means they take ready-made component. Fungi are absorptive in function, and with digestion with extracellular enzymes. As I had told you that from the host, they give out the enzymes into the host cell, and those extracellular enzymes, they digest the host material, and then that material is incorporated or taken it by the obligate parasite hyphae. Then we see reproduction. In reproduction, we see vegetative reproduction, which is done by fragmentation, Rhizopus, Mucor, and Aspergillus. Then we see the vegetative reproduction is by fission, children. It is also done by fission. Vegetative reproduction is done by fission and also by budding. Example in yeast, which we had seen earlier. And we see that fragmentation, fragments of hyphae, they detach from the parent mycelium and they develop into individual fungus. So these are some of the ways by which the fungi, they propagate. Here, as you can see in Saccharomycetes or in yeast, we see that budding takes place, and we see that how the vegetative kind of reproduction takes place by budding in yeast. We see that fragmentation is also a method where the hyphae, they detach from the parent mycelium, and the hyphae, they develop into individual fungus. Then we see that there is fission or splitting of cells into daughter cells by constriction. So that also happens in fungi. Budding, we have already seen in yeast, that from the small outgrowth from the parent, the bud from the parent cell, which grows into a new individual and it detaches itself from the parent. Then we see asexual reproduction by Sphorengiospores. We can see the Sphorengiospores here, and we can see the other one is by Conidia. So there are two methods of asexual reproduction, either by Sphorengiospores or by Conidia. And in Sphorengiospores, we see that they are motile, and they are formed within the sporangia, and we see that they also show these zoo spores, they show flagellum also in them. So these motile spores or zoospores, they are there. Example in mucor, in Rhizopus, we can find them. There are also non-motile zoospores, or those kind of spores which cannot move on their own. These spores which cannot move on their own are known as Plano Spores. Then we see conidia in the B diagram here, and in conidia there is a formation of conidiospores as an aspergillus. We can see here the conidia are being formed, and we see they are found in Ascomycetes, in Deuteromycetes, and they are produced exogenously. Then we also see sexual reproduction is there in some of the fungi, and this sexual reproduction is completed in 3 phases. We see the plasmogamy. First phase through which it is completed is plasmogamy. And in plasmogamy, the fusion of protoplasts of 2 compatible gametes where 2 nuclei come in close contact to each other. Then we see karyogamy, and in karyogamy we see that fusion of 2 nuclei from the 2 fusing gametes takes place in a diploid zygotic nucleus. And the nuclei of different or opposite strains, they get to arrange in pairs, and this pair is known as a dikaryon. And this phase is called the Dicario Phase. Through the process of dikaryotization, we see karyogamy or fusion of 2 nuclei from 2 fusing gametes. Then we see the third phase, which is completed in sexual reproduction in fungi, is meiosis. The nuclear fusion is followed by a reduction division in the diploid nucleus. As children, you had studied in class 10th also that how the 46 chromosomes, 23 come from each parent, from mother, and 23 come from the father. So there is a reduction division in both mother and father. In their sex cells, we see that they become haploid. And here in the fungus, the haploid stage is re-established in the fungi. Then we see as planogamic copulation. We see here planogamic copulation in this particular diagram, and we see there is fusion of 2 naked gametes. Can you see? There is a fusion of 2 naked gametes. One or both gametes can be motile. Then we also see that there can be isogamy in planogametic copulation. And when we talk of isogamy, the fusion of gametes takes place between such gametes which are morphologically similar or they are physiologically different. Then we see anisogamy takes place in planogametic copulation. In anisogamy, the fusing gametes which are fusing together, these 2 gametes are morphologically or physiologically, in both ways they are different. Then we see in planogametic copulation oogamy. What happens in oogamy? The female gamete egg or oogonium, which is non-motile or which cannot move, it fuses with the male gamete or anthroid, which is motile or which can move. So we see that planogametic copulation has got these 3 types of gametes. Then we come to the gametangial contact. This is the figure for the gametangial contact, and in this figure we see that the male and the female gametangia, they come in contact, and the male gamete, it migrates to the female gametangium, but the gametangia do not fuse in this. Then we see the gametangial copulation. This particular diagram is of the gametangial copulation. As children, you can see here that the fusion of entire content of the two compatible gametangia, where each comes in close contact, and this results in a karyogamy. Then we see that we come to the next one kind of sexual reproduction, which is known as spermatization. In this particular way of sexual reproduction, we see in advanced fungi the sex organs are completely absent in these, and what we see is the union of minute spores. Can you see this spores, children, here? The fusion of the minute spores, spermatia, which are the male spores, and specialized receptive hyphae here. If you can see in this particular area, this is the ascogonium, or the female part. So these two, the spermatium and the ascogonium, they fuse together. And then we come to the last kind of sexual reproducing fungi. The sex organs are not formed in this kind of fungi, and the two vegetative cells or hyphae, they fuse. The vegetative cells or the hyphae, they fuse together for sexual reproduction. Then we see heterothysm. There is another way of reproduction, which is called as heterothysm, and this can be defined as a condition where two complementary, here we see that two complementary mating types are needed for sexual reproduction. And Blacksley, he designated two strains here, positive and negative strains, and these two strains caused the heterothysm, which was a way needed for sexual reproduction. Then we come to the classification of fungi, children, and we see that the fungi, they are classified into four types, which are zygomycetes, ascomycetes, and basidiomycetes and deuteromycetes. Which ones are here? Zygomycetes, ascomycetes, basidiomycetes, and deuteromycetes. These four are there. And let us study here about the first one, zygomycetes. These are called the conjugation fungi, and the two great members of this group are the mucor and the Rhizopus. These are the two members of the group. The Rhizopus is also known as the bread mold with the name Rhizopus stolonifer. The scientific name is Rhizopus stolonifer. And the mycelium, they appear as a cottony growth on bread. The three types of hyphae that are found here, we can see in the diagram, is stolon, then we find rhizoids, and we find the Sphorengiospores. The rhizoids, they are fixed stolons with the substratum. The Sphorengiospores, they carry the columella on top of them, and the columella further carry the sporangium on top of them, and on sporangium are placed the spores that you can see here. The Rhizopus reproduces by vegetative means asexual means and by sexual methods. So we will go on further in the next video with the description of Ascomycetes, the next classification of fungi. So children, we conclude this video today here. Wish you all the best. Have a good day. Thank you.